This particular video is walking us through the PowerPoint on muscular contractions. At a fairly high speed, I recommend that you download the PowerPoint from the class webpage if you need it, so you can spend more time looking at each of these, and of course pause, rewind as necessary. So in order for us to get going, first thing that we need to remember is the basic structure of a muscle cell. So when we are looking at these, remember that we had our bundles of muscle cells all wrapped up together. Now, when we are talking about the outside cell membrane of each individual muscle cell, we call that part the sarcolemma. Sarcolemma is going to be this outer membrane, and then each of these single muscle cells are what we call a myofibril. Now, each myofibril is divided up into these separate sections that we call a sarcomere. And you can recognize a sarcomere by these Z lines that are in between them. And each of these are going to be one separate contracting section. So as we talk about how the muscle contraction works, that's what we're going to be focusing on, is each individual sarcomere. Now, again, the Z line is where individual sarcomeres connect. The entire length of it is one myofibril. The myofibril is surrounded by the membrane on the outside, which is the sarcolemma. Now, let's start looking at each individual sarcomere. If we were able to go and zoom into the thing, what we're going to find is that there are two main proteins that make up the sarcomere. You've got your actin, and actin is sometimes called the thin filament because it's basically two separate fibers that are wrapped around each other, and they make these long, skinny pieces. Now, this zigzag line, this is our Z line again. It's the outer edge of the sarcomere, and that's where each of these actin filaments are attached. In the middle of each of the actin filaments is the myosin. And myosin is like a long rod, and it's got these stiff heads on it. So we can go and look at each of these separate bands. Here's your Z line, here's the other Z line. These are your actin filaments, and these thicker bits are the myosin. Now those two pieces together are what are actually going to form the muscle contraction. So if we were to go and look at the drawing, this is a drawing by David Goodsell, and you can see that he's made this as scientifically accurate as possible. You have your thin actin filaments on the side, and here in the middle you have that thicker myosin with those individual heads. If you were to look at just one of those pieces of myosin, it looks like this skinny fiber, and there's the heads at the top. But they're kind of braided up together, and that's what's making this thicker filament, the myosin. So, looking at how these work, before we actually get into the details of the actual muscle contraction, it helps to understand what it looks like when it's relaxed, and that's this top picture, and what it looks like when it's contracted, and that's this bottom picture. Now you'll notice there are a couple key differences. First of all, when it's relaxed, the length of the sarcomere is a whole lot longer, and that's because the actin and the myosin are not overlapping everywhere. But when it's contracted, it takes up less space, and that's because this myosin is now pulled in as far on either side of the actin as it possibly can be. So as a result, the entire thing gets shorter, and as the parts overlap, they get thicker. And you can feel that on your own muscle cell, where in order for it to pull this bone forward, what it's doing is shortening this muscle. It gets shorter, and it also gets thicker because all of those separate sarcomeres are going from not overlapped to overlapped. And as they overlap, they are taking up more space inside the muscle. Now, the process of how all of this works is kind of like a Rube Goldberg machine. You've got one step, which causes the next step, which causes the next step, which causes the next step, and eventually you're going to get a muscle contraction out of this. So the trick is to kind of keep track of the steps and be able to figure out how all of the parts fit together to cause the end result, which is taking these and making them overlap each other. So let's look at how this happens. Step one, you have got a nerve, and this nerve is in contact with your muscle cell. It's almost touching, but not quite. There's an area of that membrane, remember we called it the sarcolemma, and that nerve is almost touching it, but not quite. There's this tiny little space in between. Now the nerve is going to send out a neurotransmitter, which is basically like a signaling molecule from the brain. And this signaling molecule is going to spill into this area around the sarcolemma. And we call that signaling molecule acetylcholine. Now acetylcholine is going to start to come in contact with the membrane of the sarcolemma. And that particular molecule is going to bind to areas all along the sarcolemma. So here again we have the end of our nerve. These little red squares are the acetylcholine. The acetylcholine are spilling into the space around the sarcolemma, and they are going to go and attach to receptors on the outside of the sarcolemma. And that attachment is going to cause what's called an action potential. Now later on, when we start to talk about nerves and how they function, we'll talk in more detail about what exactly an action potential means. But for this video, what's important to understand is that that action potential is basically a change in the electrical charge of the sarcolemma. So it's kind of like shooting a jolt of electricity into this. The acetylcholine gets received, and that causes a change in the electricity that is all along here. What is that charge? Is it positive? Is it negative? That's going to change. And as it changes, that's going to cause changes to happen inside the cell. So now we move forward. Again, we are looking at the top. That is a close-up of that nerve area where the nerve cell was coming in contact. The acetylcholine gets received, and when that action potential comes in, it travels down these tubes that are inside the cell and causes a special area of the cell called the sarcoplasmic reticulum to release all of the calcium it's been storing. So there's lots of calcium. It was in storage in the sarcoplasmic reticulum.
Ravenwood, please report to the bus for the Interact Club. Ravenwood, please report to the bus in the student parking lot for Interact Club. Thank you.
Okay, so the calcium comes out of the sarcoplasmic reticulum and it starts flooding the inside of the cell where these sarcomeres are. So once it starts to come in, it's going to come in contact with our actin. So remember that meanwhile we had this actin fiber. Our actin fiber is made up of a couple separate parts. Those two bits of actin that are wrapped around each other and make the thin filament, and another kind of protein called tropomyosin. And you can see the tropomyosin is overlapping the actin. On each tropomyosin is this area called troponin, and here it's drawn in purple. Now, calcium is really attracted to troponin. So, what's going to happen here is the calcium that got released into the cell because of that action potential, the calcium is going to attach to each of those troponin areas, and it's going to form what's called the troponin complex. Now, you can see in the top picture, there is no calcium bound. In the bottom picture, there is calcium bound. And the big difference between no calcium and calcium is what this tropomyosin is doing. When there is no calcium, the tropomyosin is covering up the part of the actin that the myosin is attracted to. When the calcium is present, it makes the tropomyosin move, and it reveals all of these separate areas along the actin where the myosin can bind. So before, the myosin could not bind at all. But now it's revealed, and so it can bind. And that's all because of the calcium. The calcium is all because of the action potential. The action potential is all because of the acetylcholine. So remember how I told you, one thing leads to the next thing, leads to the next thing. That's what we are seeing going on here. Now, once it has released, we are ready for the sliding filament activity. And this is often referred to as the sliding filament theory, and it's kind of describing the series of steps that repeat each other. So, for the first step here, you are going to have to use up some ATP. And if you remember from your previous biology courses, ATP is a molecule that stores energy. It is basically a series of 3 phosphates attached to an A nucleotide, and those 3 phosphates that are there have energy stored in those bonds. They're unstable. So if you pop off one of those phosphates, you can take the energy that was stored in that bond and use it to do work somewhere else in the cell. So we are turning ATP, adenosine triphosphate, into ADP, adenosine diphosphate, only 2 of the phosphate groups now. So that's what's going to happen. We're going to take the energy out of the ATP, and that is going to take this head that's on our myosin, remember that's our thick filament, and it's going to cock it back into position. So kind of like you would cock a mouse trap, and then now the mouse trap spring is loaded and it's ready to kind of move forward and do its thing, catch the mouse. In this case, we are not catching a mouse, we are catching actin. So we are going to use the ATP, and we are going to energize that head and get it ready to attach to the actin. And at that point, once the calcium has revealed those myosin binding sites, it is going to bond to that. And it is going to go and perform a power stroke, which basically means it's going to go and pull the actin closer together. We had actin on both sides, so they're getting pulled closer together. And that's happening repeatedly. So, once it has pulled it forward, more ATP is going to be used. This time, the ATP is going to be used to get the head to release the actin fiber, because it can't keep doing it if it's still attached. So ATP gets used, it releases the actin fiber, and now more ATP is used to cock it back into position. And we're up here. And now it performs a power stroke, it moves it forward again, more ATP is used to get it to release the actin, more ATP is used to reset the head, and then it keeps on doing this. And so the ultimate result is the actin gets pulled in, and the entire thing starts to get shorter and shorter. So you can see, one head is busy pulling while the other is repositioning itself to do it again. This is going to happen until that muscle cell is fully contracted or until the calcium is gone and it can't attach anymore. So, once again, our contraction overview. We started in this relaxed position. Here are all the little heads that we were talking about, all the way along here. Here is the actin that's going to have its sites revealed. Once the sites are revealed, it's going to pull them closer, 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 closer until we are in this constricted position. And now the myosin is fully overlapped and all the way at the Z-line. So that sarcomere went from being long to being shorter. And that happened everywhere along the myofibril. The entire thing went from long to short. The end result was the muscle got shorter and thicker. And as it got shorter and thicker, that's what's moving your bones. So, once again, looking at it from the beginning, way up at the top there, there's our nerve. Here, where the nerve is contacting the sarcolemma, we've got acetylcholine being released. The acetylcholine gets received, and that's going to cause an action potential to sweep all the way along here, that change in electrical potential. That action potential is going to travel down into the cell, and it's going to cause the sarcoplasmic reticulum to release all the calcium into the middle of the myofibril. As the calcium gets released, calcium is going to bind to the troponin, and it's going to form the troponin complex. When it binds to the troponin and does that, it's going to cause this tropomyosin that it's attached to move out of the way, and that's going to reveal the actin binding sites. When the actin binding sites are revealed, ATP is going to be used, and it's going to cause the myosin heads to pull this actin closer and closer together until it contracts. That's going to cause the contraction. Now, in order to relax, we need to reverse this process. So, first of all, we have another enzyme. This enzyme is called acetylcholine esterase. And when we are talking about the names of enzymes, enzymes always end in ase. And the part before it kind of tells what the thing is acting on. So in this case, there are bonds that the acetylcholine formed, ester bonds, and it's going to break them down. So that's the acetylcholine esterase. It's breaking it down. So it's a big word, but if you kind of take it apart into smaller parts, it's not so bad. So the acetylcholine esterase is going to break down the acetylcholine. Once the acetylcholine gets broken down, now we need to use up more energy to finish getting that cell ready to do all of this again. Because as of right now, the cell is not ready. The myosin is still attached to the actin, the cell is still flooded with calcium, and so it can just keep on staying fully contracted like that until we reset it. So, some energy is going to be used. We're going to do active transport and pump that calcium back into the storage area of the sarcoplasmic reticulum. So you are moving it from where there is a lower concentration out in the cell to where there's a higher concentration back in the sarcoplasmic reticulum. That's going to take some energy, and you're going to pump it, and then once the calcium is gone, at this point, that troponin complex is broken down also, which means that the tropomyosin is going to cover up the binding sites again, and now the actin and myosin can't bind. Before that can actually cover it up, though, we need to get rid of any myosin that may be attached to it. And so once again, that's going to take energy. ATP is going to go and move those two things apart, and that allows it all to kind of reset itself for next time. Once there is no longer any attachment and no ability to make an attachment, those fibers are going to slide back apart so they are no longer overlapping. And it's going to go from the contracted state to the relaxed state. So there are a lot of places in this entire process where we were using ATP. We were using ATP to go and reset the sarcolemma, pumping all of those molecules that moved in and caused an action potential. We need to pump all of that back out again. We need to use ATP to pump all the calcium back into the sarcoplasmic reticulum again. We need to use more ATP to go and get the head to come apart from the actin. So the myosin and the actin have to be separated. All of this takes energy. Now, final slide of this is talking about interference, either from Botox and botulism. They're preventing you from releasing acetylcholine, so you can't make the muscle contraction in the first place, because that was our very first step. It triggered everything else. Rigor mortis happens after you're dead, and that's because you're not producing energy anymore. And if you are not producing energy, you cannot do all those steps at the end there that took ATP to get your muscles to relax again. So when all of the calcium floods into your cells, your muscles contract and your body stiffens up. Without any ATP, your body stays stiff until things break down enough that they naturally come apart on their own. So this is the key problem here. Okay, that pretty well runs through that PowerPoint. Feel free to rewind it.